Also, we will start letting the people get in. So we'll continue with the second half of the presentation by the node or the node representative or the people in the node. And we don't need further information. We have the news from Marseille by
Yes.
Pierre-François Laine from IBDM.
Thank you. So I had initially planned to present my own work, but I had initially planned to present my own research, but I thought that it was more appropriate to present you news from Marseille. So what for people who don't know what we are doing. So I will briefly present the newcomers in the node, and then I will tell you what are the advances in terms of technologies that some of which have been transferred to the platform. So the news in terms of what additions have been made recently to the Marseille node. We celebrated last year the 20 years of the shared imaging facility, the Pixel imaging facility. And 1 year ago and this year, we as well welcomed 2 new platforms in the shared imaging facility. So the platform from the Microbiology Mediterranean Institute and the platform from the Euro-BioImaging Mediterranean Institute. And in addition, there is a new R&D team, the team of Paul Villoutré, who's developing artificial intelligence approaches to represent life, in particular development, trying to connect imaging data with transcriptomic data. So I will briefly present the 2 new facilities which join the Pixel. So the first one is IMM, so Microbiology Marseille Institute, which is technically headed by Artemis Costa, who was there remotely yesterday. They have a strong expertise, in particular in cryo microscopy, and they bring as well to our node 3D SIM, which was not present in the infrastructure. So the second platform which joined the node is the one from the Neurobiology Institute on the Le Mans campus, which is technically headed by François Michel, and which brings to our node as well approaches to neuroscience. In particular, there is a strong expertise in combining multiphoton microscopy and new contrast microscopy to other types of measurements, electrophysiology in particular. Okay, so now a few words about the node. So we provide, as you all, we provide approaches to cell imaging, including specific imaging contrast, CARS, SRS, polarization resolved imaging. I think we are motivated by this because we know that living systems are intrinsically dynamic, and we need approaches across fields, immunology, neurobiology, microbiology, and developmental biology, to tackle questions to understand how organisms grow, how they adapt to their environment, and how they are trained. And so we developed different technologies over the past years, and some are still under development, and I will go through some of them. First, I will present some of the activities of the Center for the Immunology Center on the Le Mans campus, which develops approach to look at the dynamics and organization of the membrane in response to immune in the context of immune response. One of the approach which has been developed already about 10 years ago is based on fluorescence correlation spectroscopy, and which allows to look at fine grain organization of the membrane by measuring what is called FCS diffusion laws, which relates how molecules diffuse with the spot of observation. And deviation from linear relationships is a signature of confinement from different from membrane molecules or from this underneath cytoskeleton. So this approach has been complemented over the years by single molecule localization methods, and now there is a complete software suite which is available and which has been developed in the platform and as well in the group of Didier Marguet. And more recently, the group of Didier Marguet as well started a collaboration with theoretical physicists and as well experimental physicists in Montpellier to look at interactions between molecules which are at far distance. And the idea is that upon excitation, which can be chemical or through optics, through light, molecules can interact at long distance, forming a large range electrodynamic forces. And to test this, there is here a combination of terahertz spectroscopy with fluorescence correction spectroscopy. So this is an ongoing project in the group of Didier Marguet at the CML. So now I would like to spend a few minutes to talk about development in the Fresnel Institute. And we talked yesterday and as well today about this, the increase of combination of instrumentation with numerical approaches, co-design, this idea that we don't need only to develop instruments, but as well we have to connect them to numerical approaches to model the system as a whole and as well to solve the problem, for example, in case of inverse problem. So in the Fresnel Institute, there has been over the years different developments. One which started already 10 years ago with Pierre Bon, who was there in Marseille, and then we moved to Bordeaux, and now we moved away in Limoges. We started in Fresnel to develop approaches to measure with phase microscopy structures in cells. And the recent developments are focused on measuring the phase, the gradient of refraction index, and for that they use wavefront analyzer, which is positioned in front of the camera to measure the phase of the incoming wave. And if there is a distorted front, it's possible to calculate a phase image which complements the transmission image. And because there is simple relationship between the reflection index and the dry mass, it's possible to measure mass of objects such as ear neurons, where you can see mass in picograms for all these cells. So the method can be used for measuring dry mass, but as well to look at dynamics, cell division, for example, measuring temperature locally in single cells or in tissue. And here is an example of this in complex tissues. A second example of development which has been transferred to platforms, a project that we heard about already yesterday about super resolve polarization microscopy. We usually use super resolution to locate molecules, but we can measure as well orientation of molecules. And this is what Sophie Bracelet in particular developed over the years. And here the recent development have been to make the method fast. So this is the image on the left where you see imaging of cells, and you can measure either cells live or here fixed, and do even storm, and by fast imaging you can reconstruct in 3D the orientation of actin filaments in this case. And by engineering the point spectrum function, it's even possible to look at the orientation of single molecules, not only in the plane but as well in the 3D geometry. And okay, so I changed topic now at another level, at the level of tissues. We had this morning an interesting session discussing how we can image without contrast tissues for preclinical studies. So anapathologists are used to this type of images with colors, and they want to have the same, but maybe with other types of contrast, and using SRS Raman stimulated Raman scattering or coherent Raman imaging, it's possible to image fast these tissues, and as well to have images which are similar to the ones that the anapathologists are used to. So this can be used for tissue, but this can be used as well for organoids, some of which are difficult to label when they are produced. And last development in Freinet I will mention, and it's as well related to something which has been discussed yesterday, whether R&D teams which are outside the FBI perimeter can contribute. They can. In this example, it was a collaboration between the Fresnel Institute and Toulouse, where they co-developed random illumination microscopy to image at super resolution fluorescence samples, and the method is based on speckle elimination, and by using variants of the image, it's possible to through an inversion to get a super resolved image in 2D, and as well in some cases in 3D if you scan fast the sample. So this method is, for those who are interested, has been developed in Marseille and in Toulouse, and now is in Rennes, and as well in an imaging platform in Marseille. Okay, so now a few minutes about my research. And I will discuss briefly what we are interested in. So we try to understand how tissues are shaped, how forces produce tissue flows and tissue patterns. And here are two examples, one in vivo. So this is the morphogenesis of the Drosophila embryo on the left. And on the right, something which I've been starting to work on 5 years ago. This is organoids or embryonic organoids, which self-organize and which mimic the development of early embryos. There are very different systems. One is very constrained in terms of geometry. The system are highly pre-patterned. So in a way, the cells are ascribed with some cellular properties that will deploy over time during development. And on the right, you have a completely self-organized systems where cells don't know where they are to start with. They don't have any pattern to start with, and then they self-organize to give rise to different types of tissues. So they pose different challenges. So we try to understand the mechanics of this system. In the case of Drosophila, recently we developed approaches to manipulate cell junctions. And here on the right, you see a movie where we could, in a developing embryo, we could tweeze interfaces, cell contacts to fully remodel them and to understand the mechanosensitive response of cell contacts. At the level of tissues, but using super resolution microscopy, we used STED microscopy to revisit as well the organization of cell-cell contacts. And here what we've done is to use by pairs different molecules which are known to be part of the apicobasal organization, the polarity complexes. The biochemistry tells us that there are many possible interactions. We look by STED at the localization of these different molecules by pairs, and we can remove many of these arrows which were proposed at the biochemical level, but in fact which do not exist when you look in tissues by STED because the molecules are too far apart. So by constructing a super resolution image and its super resolution map of the position of the different molecules, we can rule out some of the interactions which were proposed from biochemical studies. And this method using STED at super resolution allowed us as well to look at how actin is organized at the vicinity of the adherent junctions, and we use the same approaches to roll out some of the possible interactions between the cells. I have a few minutes, 5, 4, 3, 2, 1. 5 minutes or so. Okay, so I will finish with something to echo what Jean-Baptiste has presented this morning. A few years ago, already 4 years ago, thanks to France-BioImaging, we were able to transfer SOS-PIM from Bordeaux to Marseille. And the problematic we had that we still have, and it echoes what Jean-Baptiste presented, is how to image organoids. So in our case, we want to image embryonic organoids. How can we image them at high throughput? So what we do and what people do usually, they use epifluorescence. So on the left, you will see a 96 well plate, or not exactly 96, but many wells that we can image with epifluorescence. So if the imaging here is 2D, it's rather high throughput, but the resolution is low. If you look at these embryonic organoids, they tend to polarize. You can look at different features during this polarization, but you don't have any resolution, at least no resolution at the cellular level. At the other side of the spectrum, if I can show you on the right, multiphoton movies of these organoids when they self-organize. So you see flows of cells, which leads to the polarization of this aggregate. We have high resolution. We can follow subcellular structures. We can image in 3D, but the throughput is very low. So how to bridge the gap? So, but before that, just one piece of information about the system we look at and why it's exciting. It's because these gastrolids, these embryonic organoids, which develop in the dish, they reproduce, they lead to the different germ layers similar to the ones which develop in mammals. And they are as well, and if you keep them for a few days, they give rise to interesting structures, heart structures, somatogenesis, so vertebra primordia. And there is a lot of excitement to look at these structures. So let's come back to the imaging. So we recently, in collaboration with Jean-Baptiste and Virgile Viesnop, we started to use the GWell that Jean-Baptiste has presented. It has many advantages that Jean-Baptiste has already highlighted. And we could use this approach to look live at gastroloids. So here is an example of imaging over a few hours of these single gastroids, where we can image 20 live gastroids over time, over one day or so, with a time resolution of few minutes, and we can get subcellular details on this. So this is another example of the imaging, which shows you, at least for the ones which are close to the screen, to see the type of details we can get. Sorry, and so we can already, with this type of data, get information about the morphology, the maps of expression at the cellular level, the cell density. We have either coarse grain approaches. Oh, it doesn't work, this one. Okay, where we can compute here flows of cells and look how the polarization of these systems arise. And the next step, and this is still ongoing work, we need as well to segment the cells. So something we have started to do on SOSPIM images, but here I show you just an example of what we do with 2-photon imaging on these tissues, which at this point is better resolved than the light sheet approach. So the idea is to go to the cellular level to look at not only the behavior of the cells, whether the cells divide, how they move, how they reorganize, and how it's connected to the differentiation state of the cells. But this is still ongoing work. And with that, I'm done, and I would like to thank you for your attention.
Thank you, Pierre-François, for this wonderful tour in Marseille. So, any question? Okay. No.
Yes. It's about polarization. So, if I understand properly, you're targeting the relative orientation of the dipole of your
Yes.
absorbers. And can you do it with Raman spectroscopy?
Sorry?
Can you do it with Raman spectroscopy or on similar systems? Because you're using the fact that the objects you're targeting are oriented in your sample.
So here, the example I showed from some Sophie bracelets are from fluorescence imaging. So you need to attach the fluorescent molecule in a rigid way so that it reflects the molecular orientation of here the actin, what I showed. Could you do it with Raman? Probably not at the single molecule level because the signal would be much lower. I don't think it has been done, but this we should ask the expert. Yeah.
I had a question on your own work, I would say. You showed this nice gallery of gastruloid. So the question is how you will deal with variability from one to the other.
Okay, so I think the variability is a chance because it helps us to understand why the development of an animal, an organism, is robust. So these guys here are floating in a well, so they are free to change their shape, their boundaries, while in an organism, so there are fixed boundaries in terms of the geometry usually, as well in terms of interaction with other tissues. And I think we can, looking at variability, how the variability is reduced when you impose more constraints, we can understand, I mean, it's a way to understand better what happens in vivo. So this is the nice answer. Then you have the more difficult answer. So to say what is the variability, is it due really to, does it reflect the variability of the system itself, or something you don't master at the experimental level? I mean, I think this comes with time. So we have to understand better what are the conditions of culture of these guys. But yes, I think we usually, people in the field of organoids, they tend to consider that variability is a problem. I think it's not. I mean, it's not a problem if the outcome is robust. The variability in terms of the way the system develops is interesting per se to understand the origin of robustness.
I had the last question from your answer, I would say. This story of a boundary. So do you think there is an interest to revisit, I would say, using the similar approach, what has been done for years on Xenopus embryo, or this kind of things, or this reconstruction of
yes. Yes, I think so. So for those who are not expert in developmental biology, Xenopus has been the system from which we've learned a lot about the mechanics of tissues, and people have used this have used explants to make a simple system to understand morphogenesis. I think yes, indeed, we learn about development using these in vitro systems. They are very interesting because they can give rise to organs. But I think the way they form organ is interesting per se, and we learn from that as well. Yeah.
Okay, thank you very much, Pierre-François. So.
Okay. The Île-de-France South Node. So I will present. You hear me? Yeah. It's okay. So this work is developed at ISMO. So we are a R&D team within the node. So basically, the idea is that I would don't spend too much time on that, is to improve single molecule localization microscopy, where I think you all know how it works. But the main idea is that I want to point out, when you're doing this localization here and you try to find the center by adjusting the PSF, in fact, you only analyze the spatial parameters. So it means that any distortion due to even some small defocus or aberration will already impact your lateral resolution. So then you know that to then find the 3D information, you need to add another approach to introduce a kind of dissymmetry within your front spectrum and move from this 2D image to a 3D image And the most used techniques is still so far astigmatism. When you introduce this change of shape, will then translate the shape into a Z position when you need to have a very good calibration to do so. And then the main problem with this astigmatism is that the precision you can get, if you look at the Kramer lower bound here, the precision you can get for these techniques really depends on the position within your capture range. So if you have a typical capture range of 700 nanometers, so depending if you have a strong or regular astigmatism, you see that anyway, for example, for this configuration, you will maybe reach 20 nanometers, but on the border of your capture range, because it's getting more and more difficult to adjust this properly, you will have a precision that will drop by at least a factor 2. So you have a non-uniform precision. So then the another major issue, when you want to go in depth, so not looking at your first micron within the samples, but maybe target the nucleus or even something more complicated like tissue, the aberration you induce will get added to the aberration from your samples. So it means that the precision you will get will drop very quickly. So there are some strategy, of course, some other strategy. You can try to add some correction with some active mirrors. You can try to do other techniques like selfie. But then the idea was, can we try to use another parameter, like try to introduce a time parameter within the localization process to improve the localization precision. So the idea was, if we try to replace the uniform excitation by a structure one, like this one, so you create an interference pattern. In fact, then what you will observe for the fluorescence, because you're exciting with something that will create a modulation, now your fluorescence will also exhibit a modulation along time, okay? So it will be up and down during the time. And if you stop, then here, the pattern, and you don't move it anymore, you will see that the modulation emitted by these emitters here is at the maximum here, for example, and then decay along the time, and then drop and increase again. Whereas this one here, when it starts, it's nearly minimum and then increase, and then just by comparing the difference of phase for all these modulated signal, that will allow you then to directly find under which part of the fringes you were, if you're at a maximum or a minimum. So in fact, you use this structuration to reposition much more accurately where is the molecule. So in fact, you don't need to sweep your interference pattern continuously. You just need 3 to 4 positions, and that will allow you then to sample the modulation of your single emitter, and that will then allow you to localize it much more precisely. So basically, then you need to change a bit your excitation. You need to create interferences, and this is what we did. So you need to split your initial laser in 2 arms here. Introduce an electro-optical modulators to sweep the fringe at different phases. And then here you see, by playing with this knife edge mirror, you can change the distance between the 2 beams here. And you can have smaller or bigger fringes. And then if you do this kind of modulation, you will enhance along the lateral direction, for example, X. And then you will have to choose if you want to X, Y, or Z. So, and then have multiple pattern ready. And this is the kind of setups we build. So this is the one that we built on purpose to go to MIFOBIO. So you see it's like 30 by 30 centimeters. So it doesn't really need to be that big. You recognize here the 50 beam splitter, the knife edge mirror, the electro-optic modulator. So all this allow you very simply to create the 2 beam that will then enter the back focal plane of your objective to create the interference. So of course, in the lab, we are more interested into like improving the imaging in depth and along the Z direction. So we decided to create the interference a bit differently. So to have a beam that is focused nearly in the center and one on the side, which you see generates some tilted fringes. So it means that now when we are moving the fringes by changing the electro-optical modulator, the fringes are moving along this direction. So it means that to extract the information of any single molecule event along the Z-axis, we need 2 information, the phase information along this direction and the X information. That the combination of the 2 allows us to position then the molecule along the Z direction. So now you have also to change, of course, the detection. So that's where come the, let's say, the tricky part. So we are looking at single molecule events. So even if it looks like a SIM microscope for some of you, it's a single molecule microscope. So in fact, we're just borrowing the structure pattern for the excitation. And then if you think about it, then all these molecules, when they are blinking, they are emitting over a very short time, like let's say 10, 15, or 20 milliseconds. So if you would like to acquire this sequentially to sample the modulated emission, it will be too slow because the camera we're using doesn't have a fast enough frame rate. So what we design is we design this kind of module when we change the detection module here in order to insert an optical element, a mirror, the focal cells, that it will in fact send the image in a sub-part of the camera in synchronization with the position of the structural illumination. So for example, you see that for this first position, you will send to this sub-around the camera, and then for the following one on the other one, and this, and you can address this at the kilohertz regime. So it means that you will at least perform this 50 times, so 50 cycles of demodulation of your camera, while your camera, in fact, at the end will only acquire one image. So it means that like this, you can beat the low camera frame rate by inserting this element that can go much, much faster. And that was in fact, what we patented initially for these techniques. So how does it look like in real? So it's typically what we have. So we have this 4 different sub-around the camera. When you see we need to associate for each single molecule event is like 3 other partners, let's say, to reconstruct and then to be able to perform the analysis and extract the phase and thus the position. And then if you do so, you see that you will have this phase information that we then unwrap and then have this kind of image here. We apply the modulation along the Z direction. So what do you gain with this kind of techniques? If we look at the Kramer-Rao lower bound, so here when we apply along the X direction, so for lateral improvement, you see that, so the theory in red and then the data in blue, so you see that we typically reach a precision between 2 and 4 for the lateral direction. And then if you want to compare if you apply or not the fringe, you can see, for example, you just apply along the X direction, so you will get a precision below here 4. And then if you don't apply along the Y direction, you see that you have a precision that is indeed more than double. And we do get this 2.4 enhancement just by applying this, let's say, modulated emission. So then you have to decide which parameter or which direction you're gonna encode. And today I will mainly show you for the Z direction. And if you do then this process for the Z direction and you look at what you expect in terms of precision, you see that you should get this blue curve here for the Kramer lower bound. So you see that you have a precision that is close to 6.5 nanometers. So you did have the gain compared to the classical astigmatism that is most used so far. And then what is also important is you see that the precision you expect is like a fairly uniform one compared to which drastic change compared to astigmatism. And of course, we check that into a real sample. So here we label the cells with some DNA probes in order to, because they are emitting over a very long time. So it means you can really repeat the same localization and the same emitters and really see if you can have the precision you expect. And you see that here, that's what we get. So this is the precision following this measurement that has been performed over the first 7 microns within the samples. And you see that the precision is like quite still. And then we do have a precision that is slightly below 7 nanometers, so 6.8 nanometers. And on top of it, if you check also the lateral precision is not affected because we're not changing the shape also of the postband function. So now some results. So here we label some clathrin-coated pits. So inside the COS7 cells. So here we're looking on top of the nucleus at 4 micron in depth. So the color coded are really the Z obtained by these mod-log techniques. And then if you are looking on this region of interest here in yellow, you see that we do see that we are looking at the layer that is forming kind of a shell of top of the nucleus. And if we zoom on these 2 clathrin-coated pits here, you see that every dot is the dye and the size of the bit that represented really correspond to its precision. So we really have a nearly, it's not fully isotropic so far, but it's really a very uniform precision at least that allows to reconstruct them quite nicely. So we also investigated like much deeper. So here it's on mitochondria at 6 microns. So we label TOM22. So it's again in storm. And then you can zoom in various area. And for example, you can zoom on this one here. And again, every dot is a single molecule event localized. And you can also try to see how well you can then image the top and the bottom membrane of the mitochondria. You can measure the size, which of course contain the primary, secondary, and the precision of the techniques. And you can repeat that over the whole field of view and see that it's like really uniform. And then of course you can reconstruct this mitochondria in 3D and see it a bit better in 3D than like having a flat representation. So we went a bit further because we are targeting also a much more complex sample. So here, Abigail observed some and designed some cells embedded in collagen matrix. So as you probably know, it's highly scattering. So this is the kind of epifluorescence image you could get. And you see this is the point spread function we're working on. So they are fairly distorted, but then we just want to see if we can probe the modulation within the even distorted point spread function. And you can see that we can, because we can still get some 3D image in this kind of samples. So this development has been supported this year by the outreach call. So this is done in collaboration with Arnaud Chevrolier, Karine Monnier, and Grégory Janon in Bordeaux. So some different application. And the idea is to transform this microscope that is already working pretty well into something that is more user-friendly. So we want also to increase the imaging depth a bit further. And of course, also we want to have a much faster demodulation. We try some mirrors, but it's still too slow. So the first good thing is we confirm that we could enhance. So now we have a field of view that is 4 times bigger. So we have nearly a 50 by 50 microns. But then by increasing the speed, we'll be able to have a much higher cycles, much shorter acquisition, and also target some live cells application and do some trackings also. And then another important request by this external user was also to have a multiple target analysis. So then for that, we borrow what is typically developed at the moment in the single molecule communities to use this spectral demixing, where in fact you keep a single excitation. So it means that for us it's good because we only have then a single pattern to prepare. And then you use at the emission, you have to insert a dichroic. So it means even if you have 2 dyes that are very close spectrally, like here, Alexa 647 and CF 680, you can decide to put any dichroic at depending on what you want to privilege for the 2 dyes. And you see that on the 2 camera, the ratiometric measurement you will perform will allow you, for example, here, the Alexa 647 is 75 and 25 here, for CF 680 is 50-50. So then you have to pick the right dichroic also to allow major part of your photons to go towards the mod lock detection and not necessarily towards the ratiometric measurement. So this is what we are working on at the moment. And Abigail got the very first image last week. So you see that here we have labeled the TOM22 for the mitochondria in CF647. And the lamine B here on the nucleus in CF680, and we can see that the 2 are demixing quite well, and they are clearly in different planes and different X. So we are still working on that. And we also started to work with Karine to try the sample. So now what is nice is that we are really looking at the inside muscles, mouse muscles. So they label with Céline, the tubulin networks inside the muscle. So here you can see that they are doing this kind of funny shape around the nucleus here. And this here plane was acquired at 5 microns. This one are 12 microns. So we're actually trying, so they can make slices up to 100 microns. So the idea is to see up to which depths we can image. And we also started to work with Grégory and Tianchi. So this is focal adhesion in Drosophila embryos. And this is again, so here this one was at early age, so it's close to the cover slip. But then if you do this region of interest, you can see how all the proteins here, the integrins are arranged, and you can see how it varies in terms of Z position. So just to conclude now, so you see that introducing this temporal parameter really had another way to reveal the position of the molecule, and in particular to benefit of a uniform precision. So you can think of applying into any, so X, Y, Z, even orientation, for example, or lifetime. So it can be generic to any other parameters. And we are still working. So I've already talked about a few things we can do to enhance. So we can also play a lot with the tilted and the size of the fringe pattern, also to play on the resolution we can target. Because we also have a system that encodes the Z very nicely, we also want maybe to increase the volumetric imaging. So try to get in a single acquisition a much larger depth of field. This is what we try by having some phase mask that extend the focus. And you can see that you can have up to a factor 5 enhancement of focus compared to without mask and with the mask. So you can extend the detection capability of your microscope. And you see here the two images, the one in red and the one in blue, clearly exhibit a difference of Z capability. And also the motivation to go fast with this new module is also to be able to, as Lydia says, there are now more and more self-blinking dyes that are existing, and we are targeting to use them. So you can see here that this one was targeting tubulin, and they have been taken at 5 minutes apart. And you can see that we can probe now in super resolution, the structure evolving. Here is going to this direction, and here it's this direction. You can see them really like acting in live at the nanoscale. So I just want to thank the team. So this project was initially developed during the PhD of Pierre Jouchet, and now it's mainly the work of Abigail. And then I want to thank all the collaborators, the supports, and I thank you for your attention.
Thank you, Sandrine, for this very nice presentation again. Question? Yep. No question. Come on, we can have a question, no? We have plenty of time. Yeah. So the last thing you showed, it was with tubulin, I think. So I didn't get exactly how you managed to. Sorry for the question. I'm not an expert. How you get this?
So here it's a classical, let's say, 2D super localization image, like a kind of a storm one, but in live cells, because these are self-blinking dyes. And the only challenge here is to acquire much faster than the image the cell is going to move, let's say. So that's why we need to go fast to have a kind of detection that can go much faster if you want to do 2D and 3D. The idea is if you want to acquire with a smaller photon budget. So if we have here, we can benefit of let's say 2.4 enhancement. So it means that we can reduce the acquisition time very drastically. So we can position events much faster. So that's another goal if you want to address live cells structure in live cells.
Okay, thank you. No question in the room. Okay, so you can still discuss after. And thank you very much for Sandrine and all three speakers. So I will leave the room to.
So, so it's my pleasure to introduce Yanero. We know each other since a long time, I would say, although we are very young, both of us. And actually, Jan has been really involved in different, let's say, in infrastructures, research infrastructures. So he's working at the IGBMC. If you don't zoom too much, it's very like Strasbourg, but it's not in Strasbourg. It's in Ilkirch. He's responsible for the clinic institute for mouse clinic institute. So he's also the responsible of Phenotyping, which is one of the research infrastructure on mouse. He's also the responsible of Selfedia, which is the French national research infrastructure on animal models. Huh? Okay, good. And so he's also very involved in the infra-frontier infrastructure, which is quite old actually. The infra-frontiers was 206. at the European level. And so today we'll, it's not the title that you give. Okay.
No,
So,
yeah, but I will show you.
okay, fine. Thank you.
Okay, so thank you very much, Jean, for this kind of introduction. So I have too many arts, I know that, but I'm really excited by the work I'm doing at different levels. So yes, okay, I will try to speak in the mic. Okay, and today I'm going to introduce you the International Mouse Phenotyping Consortium, which is in fact a big network of centers which are spread all over the world in order to simply create a comprehensive catalog of mammalian gene function. So it's quite ambitious, as you can say. But this story started almost 20 years ago. We were in 2002. We had a meeting, both in the European part and the US part, with many colleagues working on mouse genetics in order to see what we can deliver in the future as a community, you know. And we thought that we were really ready to go and to create a null reporter allele for every gene as a foundational starting point for functional annotation of the mammalian genome. This was the original goal. And we have two key principles that we are still taking into account. Is first to use a single inbred genetic background. So this is very technical, but this is for us, and to make those resources open and available to anybody that will require access to the animal, to any cell or samples, and to the data that will be collected through the program. So it was quite ambitious, and we start with several funded projects at the European level in order first to structure or concept, and then to also to fix some critical issues. The first one was really to fix and develop a comprehensive set of standardized operating procedures to investigate mouse as a model system for different areas and domain of physiology and cognition. This was the Eumorphia program. Then we moved and we generate a series of alleles through different initiatives which were funded either in Europe with UCOM and then UCOM tools, or in US with COMP for knockout mouse project, very easy to understand, or NORSCOM for the Canadian version of European UCOM project, in order to deliver a set of alleles that could make knockout for every gene in the mouse genome using the technology that we had at that time, which was mainly based on the embryonic stem cells and conditional gene activation. And then we had a preliminary, let's say, tentative event to do this type of analysis on the large scale for 500 mouse lines that were selected from the resources made here, and using the protocols that were defined in Eumorphia. And later on, with the success of these stories, we decided to build this international mouse phenotyping consortium in order really to make a systematic inactivation of every gene in the mouse genome and to do the comprehensive analysis of the phenotype of those mouse mutant lines. And of course, in 2015, we had to consider new technologies, that's a CRISPR-Cas9 techniques that you may know that are really pushing and giving us tools to engineer more quickly any mouse models that we may need to generate. And in 2021, we were able to celebrate our 10 years anniversary. And we are almost ready to achieve one of the goals of our projects. The first one is to generate a mouse null mutant for every gene, to give a comprehensive phenotype for each of those mutants using a standardized pipeline, to determine the baseline for mammalian gene function, and to get insight into the genetic basis of disease. And this is not something that you can do on your own laboratory. In order to achieve this type of goals, you need to bring together a lot of expertise and competencies. And this was done by this global network, and you can see it's already spread all over the world, with people in Asia, in US, and in Europe. And we are all contributing with the same perspective and the same vision to deliver this resource to the scientific community. So we have already a unique list of protein coding genes because the sequencing of the genome is known for years now, even though most of them are still unknown. And we had defined a unique, or at least two different unique standardized IMPC phenotyping pipeline, because when we start this analysis, we were facing a difficulty that we were never envisaged before, is that almost 30% of the genes that we inactivate in homozygotes are leading to lethality. So we had to develop a more specific embryonic phenotyping pipeline. This is what we did here. I'm not going to detail that. And we use different type of technique in order to document where this development is blocked. And we have also taken into considering the effort done in Eumorphia and Modic, made a comprehensive adult phenotyping pipeline with a reduced number of animals that could be studied and with a very good power to detect any phenotype, if any, is happening in those mutant mice. So we have basically those two mouse models. We have defined a series of procedures. It's too small to read, and which are attached to different biological systems and cognition systems that you can follow. What we have been facing is that we only face no phenotype for 6.7% of the mouse types that we have studied so far, which is very low. And this is a phenotyping pipeline without any major challenges. So, of course, there are genes for which you need this type of challenges, but we were not able to do that in our project without impacting the further tests that will be done later during the life of the animals. And we were able to document most of the phenotype. And as you can see here, most of the genes have more than one phenotype in there attached to their inactivation, which is quite interesting. So pleiotropy is really something which is common. And this may also help you understand the multi and comorbidities that you are facing when you have the genetic mutation in a gene of interest attached to a rare disease, for example. So the novelties that we found, so first, I already told you about that, there are 35% of homozygotes that are embryonic lethal or subviable, so leading to death just after birth, so it's a bit more than 30. So pleiotropy is a rule, so most of the genes have not a unique function, but they are active in different type of system, and this is quite informative. At present, we have done almost the analysis of almost 5,000 genes, and I will show you that we have much more now available. This was a paper for 2021. We have also unraveled a new function for disease pathophysiology with candidate genes for genomic medicine. And when we compare the data that we had at that time for more than 3,000 genes, we were able to report new phenotypes that were not found in already a mutant which has been studied so far, which is really important because it means that even if you are doing your knockout at your own laboratory, you are not looking at all the pleiotropy and all the effects the mutant line could have, because you are focusing in most of the cases on a very specific question. We have also an impact on other major specific domains. So we have been working on genes which are controlling the hearing, genes which are also affecting the sexual dysmorphisms, leading to now to consider both male and female in all the studies that you need to do using animal research. We also identify genes involved in metabolism, which were not found before or not known before. Genes which are essential between human and mouse, which are also implicated mostly in rare disease. We have developed also machine learning for phenotyping of inflammatory nociceptive behavior in mice, which was about pain research. And I will introduce you a recent paper that we have done regarding cardiovascular gene involved in cardiovascular disease, both in mouse and in human. And I will talk later on about this last paper. So we indeed deliver recently a new set of genes which are involved in congenital and structural heart defect. And we were really keen on seeing that from the 705 genes which have been found in our more than almost 4,000 IMPC mutant lines, there was 176 which were known to be cardiac genes, so involved in cardiac disease. But almost 300 were still unknown and were attached with a cardiac defect in those mutants, having very specific and very detailed and precise phenotype during development, or having late phenotype, for example, if you look at ECG. I have no time to go into details, but if you're interested, this paper is out. We have now delivered resources. So the last release is from November 2022, where you can find the phenotype for almost more than 8,300 genes. It's a bit more of mutant, a higher number of mutant lines which has been studied because there are some replication studies and other alleles which has been studied. And for a number of almost 100,000 phenotypic calls. And this is accounting for a bit more than 73 million of data points and 730,000 images. That goes from X-rays to embryonic images or a GCG. This is a description of the phenotypic annotation. I have no time to discuss that. There are certain topics in which we find much more defects than in others. This also depends on the number of variables that we are scoring in those different types of phenotypes. The phenotype annotation is quite good. We have a developmental defect and also defects which are appearing during early adult or late adult onset, which are important for the biology in human. We have a progress in making these mutant lines available to the community with all the release which has been QC validated and validated with all the data available. And how you can mine those resources, you can go to the website, which is mousephenotype.org, and go and select either for your gene of interest or for the phenotypes. For gene of interest, for example, this is a case of DIRC1B, a gene that you may not know, but doesn't matter. You have here a small vignette in which you can see in orange all the areas where there was a phenotype detected with our standardized analysis, which is also compensating for multiple testing, of course. There are areas where you have no phenotype at all, and areas where you have something maybe to look more or which have not been tested, so in gray. Of course, this is quite a small vignette, but if you want to have access to the raw data, you can go down to this level, and you can get them through the different phenotyping calls, or you can get them as a raw data set and reduce the analysis if you want. You can have also access to a multiple of images which has been captured for this, for example, for this mutant line. I have no time to discuss images that could be X-ray based or also the immunophenotyping for the FACS analysis. This is a type of, sorry, I missed. If you look for phenotype, we are using a standardized ontology in order to help people to understand what the phenotype is about. And this is an ontology which has been made in a relation with a human ontology. So here, for example, you have a phenotype which is called abnormal circulating HDL cholesterol level. If you look at this, you can find that there are only 4%, a bit more than 4% of genes which are been tested, which have shown a phenotype in this with this type of phenotype, counting for about 288 genes, whereas 6,000 were tested. And you can get access to the ideal HDL cholesterol level, which has been measured in the different centers, and you can appreciate here the variability of the measure. Which is something which is quite striking because we are using the same protocol, but of course there are specificities from each centers that makes these differences. And here you can see, for example, the normal distribution of the samples, of the value of the sample of HDL cholesterol level by clinical chemistry. And you will see that you will have some mutants which are raising up around the side. But if you go for other parameters, you will have similar effect, and you can see the effect in females and also in males. So that's quite interesting. So the pro and cons of future data mining. So we have detailed all the standard operating procedures that should be really carefully defined because this also influences the results that you can provide. And we have to revise those subs because we have sometimes to improve. We have new materials that need to be put in place, which are much more sensitive. So it changes also the data and the metadata description. We have also some slight difficulties, as I show you, to standardize all the subs. We have small variation per center that could be due also to the nature of the housing condition, which we are not completely controlled because the diet for sure and the bedding are not the same in US and in Europe. And we have to use for that a reference data, but we are always doing the comparison with our mutant line with control, which are going through the same pipeline at the same time period. We are now using all these images which has been captured in standard operating procedures in order to better do our data capture and analysis. So we are reusing now AI-based system in order to monitor, for example, ECG, and we have much more very powerful now detection of cardiac abnormal rhythms and different size of the QT and different time, specific time of the cardiac rhythms that are very interesting to look at. And we are doing the same now also for X-ray based analysis. And there is a major gain in mining this resource because you have access to a lot of data for your reference strain, and you have also you can consider to use a lower number of animals for research for your own purpose. And as I mentioned just before, there was a group here who just mined the data set in order to identify new candidate genes for ciliopathy genes based on the phenotype and the protein-protein interaction, which are kept in other databases like STRING. So they did that, and they identified a series of new genes which could be involved in ciliopathies, and they are looking for into more details. The publication based on the IMPC resource is quite exploding at the moment. We have more than 4,700 references which have been using the resource, either the models or the data, which is quite good. So there is an added value to the project. And because we have streamlined also the cost of the analysis, so it's a nice way for people to have access to raw data set and to new genes of interest, for example. So going forward, what are our goals for the next decade? We would like to achieve and complete the most null resource. We are far from that at the moment. I will show you where we are. We would like to design and produce a genome-wide mouse and resource for also human disease-associated coding variants. We would like also to undertake a pilot program to design and generate mouse strains that model genetic variation in the non-coding genome, which is, as you know, one of the big part of our genome. The coding is only 3%. And we would like to strengthen our collaboration with, for example, here, clinical scientists or human genetic consortia in order to tackle and to help for understanding a rare disease, for example. So at the moment, we have done more than 13,000 genes. Not all of them are in the repository at the moment because the data should be qualified. And there are still a series of genes for which we don't have any mouse models. So there are genes which are really with human orthologs. These are the first ones that we will targeting. So it's a bit more than 3,000. And there are genes which are specific to the mouse, which are not going, we are not going to consider first. And what we are going to prioritize at the moment is really the dark genome. So those genes for which we don't have a lot of information, and there are still so many, still a few in the genome, still about 1,000 which are left in the genome. So we aim to deliver that by the end of this of the year 2025. So you will see that or not. And in the meantime, we are going to design and produce this genome-wide mutant strain resource for human coding variations. So this is what we are going to do to enhance the studies of rare disease, to validate the candidate pathogenic variants, and identify disease mechanisms. At the moment, we have done 348 mouse mutant lines all over the world, and we are aiming at going for 1,000 in the next 5 years, so for 2025 now. And we have developed a series of new pipelines in order to be more, let's say, close to the disease. So taking into account the IMPC phenotyping pipelines, the adult one, we have developed additional pipeline. Here are examples from the Czech Center in Prague, where they have developed a pipeline for behavioral phenotyping, energy metabolisms, developmental disease, and cardiovascular screen. So these are technologies that we are implementing at the moment, and we have similar pipeline at the Moscow Medical Institute too. And the other point is to go for the non-coding genome, for which we don't have a lot of clue at the moment. There are many different types of elements in non-coding elements that we are looking at. Non-coding RNA, microRNA, gene regulatory elements, tRNA, ribosomic RNA. And this is something that we will capitalize with other initiative which has been funded by the NIH, such the ENCODE programs, the Encyclopedia of DNA Elements, the IGVF, the Impact of Genomic Variation on Function Consortium, which is really tackling the non-coding genome, and the morphic resources which are now built by the NIH to make a knockout for every gene in human iPS cells. So this is where we are at the moment, and this work would have not been possible without the involvement and the commitment of all those people which are spread all over the world. I'm very honored to be sharing this network at the moment. It's a full open science-driven project, which have quite an impact, I think, for the future of research in biology. And I would like to thank you for your attention. And if you would like to know better about this project, you are welcome to attend the next meeting, which will be in Oxford in July next year. And thank you for your attention.
Thank you, Jan. Well, any question about the work of this consortium? Well, I know we are very far from the in terms of technologies, but there is one question over there.
Sorry. For example, you showed the HDL quantification on 280 genes. I was wondering, can you do reverse genetic to look at other phenotypes on the same gene? Because since you don't have the same response for each of the mutants, you could have a kind of linear response on other phenotypes. So I was wondering, can you cross your data on several phenotypes like?
This is currently ongoing. When we tried to do that, we had lower number of genes, and it was not easy to make hierarchical clustering, for example, of genes which are common phenotype or which are partly the same effect on certain type of areas. So I think it answers your question, no?
Yeah, partly.
So these are things which are ongoing. I think we need to reach a bit higher number of mutants at the moment. For example, we don't have all the pathways which have been mutated, for example. So we need to have that as a kind of scheme to see whether or not we are looking at the same influence and the same consequences for all the mutants in the same pathway.
And in the database, do you put only the things that you have done, or can you add some extra publication so that we have a bigger database with everything?
So at the moment, we can only deposit the data which are closely linked to SOPs. Because these are the SOPs which drives, in fact, the variables and the metadata which are captured and analyzed for the variables which are analyzed. So you need to deposit the data SOPs. So we cannot, for example, enforce people to have specific SOPs. They can use ORS. Okay, so that's the first point. The second point is that we have also specific smaller program tackling, for example, infection, and they have a dedicated page. They should have soon a dedicated page for these results, to explore the consequences of challenges like infectious challenges on certain number of mutants. But they will be selected because we cannot do that. We cannot run this analysis on a global scale. It's too expensive.
Other questions?
I had one. Thank you. So it was written really small for the phenotyping workflow, but I had the impression that it's mostly like at the macro scale of physiological behavior that you are phenotyping. Do you have any? No, it's not bad. We could not see behavior or measurement of organ size, I guess, or things like that, or.
We have that in the necropsy analysis. We have all the weight of the organs, for example.
Yeah. But then my question was, what about like cell biology or proteins, whatever? Yeah.
We haven't done that. So there are some pilots currently running with proteomics and transcriptomic studies, which are very interesting, but it is so expensive to run that. Now what we are considering is to go for single cell analysis of certain selected organs. We have been dreaming of that. Yeah.
But for example, do you think like what is developed in FBI, like for example, doing developmental mouse watching, whatever, could be of any interest for phenotyping, or yeah?
Yes, of course. But this is very, very specific and very, very specialized. But of course, for certain, I think we should do that. So here we are doing a comprehensive first baseline analysis. This is what we are going to deliver. Exactly. It's a kind of clinical evaluation as you do if you go to the clinics around the age of 50, something like that. And then you have a complete checkup. Okay. Of course, if you go and you want to go into more details, you can do that, but you have to do for very specialized and non-high throughput tests. And this is something you can do, of course.
But to come back to the question of Lydia, if somebody wanted to feed with other data, then
yeah, I think we are open to that. If you are willing to fit, and if you think that you have a test which is can be put on the high throughput level, I will be happy to consider it and to discuss it with you. Yeah. We have a lot of also histopathological data sets that we are really, really willing to explore more than we do at the moment. And there are a lot of new insights inside these data sets.
How is it working? I mean, I understand the overall things, but how do you see the complexity of the IMPC in the near future? Because for what you just put,
it could be a never-ending story, but we are willing to fix it with the completion of the null resource. That's the first point. Then we'll see if we need somebody, if we need to do something else. The human variation is also another topic of interest, but there are other alternatives. We have been discussing a lot with other model organisms which are used to do that. Zebrafish, but also cellular systems. But of course, it's a matter of interest and a very specific focus of phenotype. Yeah.
We have to finish. Okay. So thank you again, Jan.
Thank you very much.
So it's my pleasure to introduce our last speaker. John Eriksson is Director General of the European Infrastructure Euro-BioImaging, and he will close today's session with a presentation with a very inspiring title, France-BioImaging and Euro-BioImaging, the sky is the limit, but the resolution is not. So John, the stage is yours.
Thank you very much. And thank you for the invitation. It's been delightful to be here, and happy birthday. I think the three of us who are here from Euro-BioImaging headquarters, we have been very impressed with what we are seeing, so keep up the good work. The title here indicates that I'm going to talk about the future. And I'm going to talk about, first of all, that it's difficult to predict the future, but also that when it comes to the future, it looks pretty bright. And it's not about technology that is limiting, it's what we aspire to do, what is our ambition. That is really the limiting factor, and I don't think that should be a limiting factor. I think many of you recognize this picture, which is from the Space Odyssey 2001. And if you haven't seen the movie, please watch it during the Christmas. I think it's a good movie. There's a human predecessor who is inventing a tool there. And that was a big revelation, and that's the topic of the movie. So little did he know that some maybe 1 million years later, there would be a guy, a draper in Delft, Anthony van Leeuwenhoek, who discovered that the ape man is made from small things called cells. Obviously, then lots of things happened. Anthony was a forerunner for the technologies we're working with, but he had no idea. He couldn't have any idea. And that's only 300 years ago. about what we are discussing here in this room and what we have seen in the presentations. But if we compress the time perspective a little bit, we cannot have, it's very difficult for us to have an idea what's going to happen in the next 10 years. And I thought I would illustrate that with a few slides from my own work as a scientist, because I'm a professor in cell biology. Did my postdoc in 1990 in Chicago, and I have been working with intermediate filaments. So this is curiosity-driven research. It's about a gene family which is very big. It's the third big cytoskeletal system, and we don't know why we have so many of them. So we have many genes. It's one of the biggest disease families in terms of human mutations. Each and every body tissue has its own intermediate filament. One of our favorite proteins have been vimentin, and I'm going to show a few pictures which are illustrating just the development of microscopic science from my own perspective. These are that are in different tissues, and you can see the broad spectrum of their phenotypes. This is the phenotype of vimentin in a fibroblast, and this is actually one of the first confocal images I took, or my group took, this early 90s. So I had been working with wide field microscopes up to that time, really nice instruments, wonderful resolution, wonderful images, pretty stuff, but you only go so far with wide field microscopy. So the first confocal picture I saw was a revelation. In those days, it was considered that intermediate filaments, just like hair, skin, and so forth, are stable structures, not like microfilaments and microtubules turning over. Then came GFP, came the FRAP technologies that all of you are familiar with. And let's see if we can get the FRAP to. So here's a FRAP movie of intermediate filament proteins. This is vimentin. This is somebody's picking my attention. So one of the first FRAP experiments we did. And you can see obviously that these are turning over almost with the same pace as microtubules, and they're moving and so forth. So then when it comes to the structures, I happened to land in an organization in Turku where there was a certain guy whose name was Stefan Hell, who was working in the same corridor as I was. And my group was giving him cells. Little did we know that he was going to be very famous. This was in 1994. And if I would have been digging a little bit into what the physicist is doing, I would have had very nice papers. But of course, we became very good friends, and a little bit later, this is the first STED image of our structure, which looks like this. So you can see that now the confocal image has changed completely. So there's a lot of detail here. Complete revelation for myself and my group. Now you can start doing mutations. We show that this turnover that I was showing with the FRAP experiment, that's due to phosphorylation. We identified the phosphorylation sites. You can examine them in very high detail with the super resolution. This is all familiar to you. We saw wonderful super resolution images in this afternoon. And what you can do now with super resolution, totally amazing stuff. But then all these experiments, they go along with proteomics. So you have combined usage of different infrastructures. Okay, here's another experiment. Early on, we observed that the Vimentin knockout mouse has a phenotype which is really striking. They do so poorly with wound healing. So they have a little bit similar wound healing as in diabetes, basically ending up with chronic inflammation. And we have been digging into this during the past 15 years. And the short conclusion is that everything goes wrong in terms of wound healing, including the immune reaction. So this is an old picture of how immune cells are invading the inflammatory tissue. And this is in 2003, and it was considered that they are going straight through cell junctions, and there is no other way. So with Sirpa Elkanen, who is an immunologist working again in the same building as I am, she was seeing cells that they seem to be going straight to cells. And then we were using our favorite protein to check whether this would be true. And I don't have time to go through the real data, but here you see one image showing that the immune cell, which is in red, is really smack in the middle of the intermediate filaments, which are in the HUVEC cells here. And we can do the stuff that one could never have imagined when I did my postdoc. This is now, we're at the turn of the century already, into our century. We could do viral transfers of tagged Vimentin into HUVEC cells. That was quite an advancement. Here you can see the border of the HUVEC cell. And now you can see that the guy thinks that this is a T cell, thinks that there's an inflammatory tissue, and goes straight through the cell. And moreover, we could show that Vimentin is needed for the process. Basically, rewriting that paradigm. And now we have the paradigms, the two different pathways are possible. We discovered when we were publishing this that somebody had discovered this already in the 1960s, but nobody believed them because that was considered to be an EM artifact. Okay, what else is going wrong with the wound healing? Again, I don't show you the real images, the live cell images, but this is an assay that many of you are very familiar with, scratch wound healing on a plate. And you can follow by automated tracking the cells. And this was for my group and myself totally cool when it came. High throughput, you could do lots of different things. And when I was doing the postdoc, one would have done this manually, if at all. Super cool. Then, what is underlying the problem that we see in the knockout mouse, that the cells get lost? So there's a problem with the migration. That is one of the problems with the wound healing. What is that due to? We checked the focal adhesions here, labeled with Paxilin, and we can see that there sort of seems to be a difference between the wild type and knockout, seems to be a little bit fuzzier, whatever. And nowadays you can analyze this with very high detail in high throughput systems. So you can see when the red is at the same place as the blue. Red is in the beginning of the experiment, blue is in the end of the experiment, meaning that the focal adhesions are aligned. And the higher is the index there, the poorer they are aligned, basically meaning that the focal adhesions are wobbling. And that is the reason why the cells get lost. So again, another reason for why the wound healing is doing so poorly. Super resolution microscopy, along with actually experiments at also Janelia research farms at the Advanced Microscopy Center, show now, this is unpublished and still in progress, but show that vimentin comes smack in the middle of the focal adhesions. So we believe that we will be able to rewrite this nanostructure a little bit. So that was a detour in our own research, and all things that I could not have predicted in my wildest dreams in the beginning of the 1990s. And that's not so long time ago. And all of those experiments done with one common denominator. And the common denominator is that they were done in core facilities with a lot of expertise related to not only the machines, how to use them, the probes, the best practice, and so forth. No chance that our group, especially being in a relatively small university in Finland, could have done that by ourselves. And so this business is still out there. And obviously, you're working to meet those needs. Covering the needs of this imaging revolution that we have seen, which stretches from molecular things into clinical things, and increasingly we will see this continuum from molecular cellular imaging into clinical imaging, and hopefully these fields will come together in reality. And that is really the whole idea of Euro-BioImaging, from poor access to open access in a continuum, and also in a continuum with other infrastructures. There's a need for imaging. There's a statistical survey from Finland where imaging, almost 100% of the scientists are indicating that they need imaging for a publication. They also obviously need other infrastructures. But that's promising good for our business. And here's the same slide that Antje showed yesterday, indicating this is our organization. This is the organization where many of you are also part of and where you are serving. 35 nodes, and look at the number here, 173 sites. That's because many of the nodes are divided on many individual sites. And in one city, you can have quite a few sites actually in one city. Okay, so then we have more than 90 technologies, and you can see the dots there. Those are the cities around Europe. And if we think about the start of Euro-BioImaging, it started the way that France-BioImaging started. So you had national imaging centers that were highly professional and had a high degree of street credibility already in providing open access. So then originally the idea was that, okay, a little bit more European funding and magic will happen. We will have this platform of European imaging excellence. Now, I think at the outset, just like here, just like in Finland, in other places of the world, of Europe, the prime argument was for this additional funding that, okay, you get the Euro-BioImaging stamp, which of course is nice. But the thing is that now you have 173 sites. And we were on the panel of nodes meeting just recently, which is an organ for the nodes to meet, obviously. And the enthusiasm there, contrary to many of the previous occasions, was really high, that the nodes and the people there start noticing that, wow, we are collaborating with an amazing structure, absolutely astonishing. The know-how behind this in terms of possibilities for training, data, funding, exchange of experience, what have you, are truly unbelievable. So I think that's for all of you to employ. So just remember that there are lots of friends out there, they're thinking exactly as you are. They have the same ambitions, the same thinking, you're speaking the same language in many different accents, but you understand each other, you know exactly what you are talking about. Okay, let's look into the future now and what it offers, and let's look at in the short term first. What we have at hand is Horizon Europe. You have all heard about this, but this is something worth paying attention to. If for nothing else, you can see the number there at the top. And that's a high number because this is for science. And we should bring back some of this bacon. Sorry for the bacon expression for those who are vegetarians, but you know what I mean. So basically you have these different pillars, and here you have is research infrastructure. This is us, and you have 2.4 billion just for the research infrastructures. And some of that money we also already have in the organization. Then in the middle, you have pillar 2. There you have the missions. And in the missions, you have cancer, you have health. You have quite a lot of funding for health. There's 8.2 billion. Also quite a lot for food, bioeconomy, agriculture, and so forth. We already have projects in those, but then the big thing is the mission part. Before we go to the missions, here are the current projects that Antti already showed. And for you to pay attention to, those who are engaged with our nodes, is this part acutely, because this is 1.4 million euros for transnational access projects. And it's with a relatively high burn rate. So this should be used within, let's say, 3 years, ideally, and we have chances for top off if we manage to spend it fast. Our user projects are in average quite inexpensive. They're in the order of maybe 5, 10, 15,000. So you can imagine that with 1.4 million euros, you get quite a lot of user projects. So we better make sure that we spend those. Isidore is the first one. It's already out there. You get the information on that. And if you want the information, just make sure you get in touch with us. We have quite a lot of projects there in the corner related to data management, which I will not go into any more detail, but that's something, again, for you to pay attention to. I will talk about AI for Life in a moment. Then you have the technology projects, which are for technology development, and I'll take those also. So here's the mission slide. And again, you may have seen these slides, and they may be a little bit of a turnoff because there's so much stuff, but we should pay attention to this because there is really funding here for us to catch. The cancer mission is a big one. There we have 400 million euros. And then we have the climate change, healthy oceans. They are also very big, but this one comes very soon. If you have any chances to engage with the cancer mission, the first project there is Understanding Cancer, UNCAN. If you have any contacts, any initiatives, anything, please let us know. Please let me know. We're trying to work ourselves into that project, but it's a big field and you need to be really actively playing in that field. Technology advancement is one of the things that we should do. And basically, when we think about any invention, some of you are doing that, inventing new technologies. It takes a long time before they get into real applications into turnkey technologies. So this is one part of our business to facilitate this. So there are these tech calls. This is the first one which came out last year. And we have now 3 of them. And Antje was presenting them already. We have for small-scale continuum, for underwater observations, for also EM. And if you're interested in this, please engage with us and with the projects. But then there will be new tech calls coming up in 2024. If you have ideas in this respect, please get in touch with us or with the France-BioImaging management team. And we have lots of collaborations with the different infrastructure organizations. So you have the chances of, first of all, having projects together with infrastructure organizations, but also planning collaborative projects. And then 3 projects as a heads up. We have one which is called a Synergy Grant that comes very soon, where the idea is specifically to combine the services of different infrastructure. The second one is interesting, I think, for many here, because it's a co-funding pilot, meaning that the Commission funds 70%, and you need to come in with 30% of own funding. This will also, if this works out, this will be a fantastic development, and let's see how it goes. Final one is strengthening bilateral cooperation with Latin America. If you have any interest in that, then let us know. There will be also an Africa program coming up in 2024. If you have any engagement with Africa, please let us know. Okay, the final few slides is about the limitless sky and also the unlimited aspect of our available technologies. We're rewriting our strategy. Jean Salamera had a nice presentation in the beginning on the FBI strategy development, where you can identify a number of things in the basic outset of when we are writing our strategy. So there are the usual suspects, of course: secure funding, strengthen the visibility of Euro-BioImaging, the brand and recognition. I think the recognition of science, you have all noticed that it's not doing so well. I think all of us could do a better job in telling the story of the great stuff that the scientists that are working with us are doing. And so please help us telling that story, but also tell the story of Euro-BioImaging. So whenever you are dealing with somebody, please let them know that Euro-BioImaging is super cool. I was on this seminar, I heard all this stuff. You should pay attention to Euro-BioImaging. We have the economic impact, foster communities, foster and grow Euro-BioImaging node community, and then increasing engagement with node communities that is happening as we speak. And then this one, adapt to societal changes. There's a lot happening right now. How should we take this into account? If we have that last aspect that I mentioned, how to adapt, what are we adapting to? I think this is something all of you are thinking about every once in a while. What is going on? Because there's so much going on, both positive and negative. For sure, for our technologies, there will be more details, more information, more dimensions, we will see that life sciences become increasingly data intensive. We go from averaging to single molecule analysis. And then this will put data management and AI in key roles. I can predict, and we can predict, that large-scale infrastructures will have an increasing role in modern science. And research infrastructure organizations will progressively become powerhouses of science. Then what is the confusing stuff? Different needs in different countries. In some countries, we have shrinking populations. In some, we have growing, aging versus young populations. Personalized medicine in our rich countries with sophisticated technologies versus treatment of really, really super poor populations, incredible poverty, incredible problems. And hopefully we can help them. Extremely expensive and advanced multidimensional instrumentation versus low-cost, mobile, and versatile instrumentation for those that are in acute need with very limited resources. So the last slides before I finish are related to the aspect of data, which obviously we all have these wonderful machines in our pocket, and in the early 90s we couldn't imagine that they're doing the things that they are doing. So this is AI for Life, a project that we recently kicked off, and this is something you could pay attention to if you're at all interested in artificial intelligence, because it's pretty amazing. And I don't know if it was hitting France the way it was hitting Finland, but there was this free tool, AI tool, that became available last week. And it made big news in Finland, and I think in some other countries. I was trying to look at French newspapers, but I didn't find so much, but maybe it's a language issue. Anyway, so I played around with the tool before I came here. And I asked the tool, how will microscopy look like in the 2030s? And this is the answer. I mean, this is the exact formulation. So first of all, the whatever, it is quite good formulating language. And it has answers that I think many of us would give. It's hard to predict. There's a likely continuation. Additionally, there's a good chance that new imaging technologies and techniques will be developed. And the positive thing is that the artificial intelligence is concluding that overall the 2030s are likely to become an exciting time. That sounds nice. What is the future? I had only two more slides. What is the future of AI for microscopy? And again, you can, I don't read this loud. You can read these comments. I think these are pretty good predictions. And the prediction is that amazing things will happen. And it's so obvious. We saw some signs of that, but the true use of AI in our science is still to be really happening. Okay. So then I asked the AI to write a poem about microscopy. And I haven't edited this. This is copy-paste. And it's quite beautiful, actually. So First of all, it has quite a lot of contents. It's not only empty words. With sharpness and clarity, it reveals the hidden. That's our work. And it's a vital tool for science. So I think these are all aspects. Sorry if I went a little bit over time, but these are all aspects for us to pay attention to. The organization that I and we are representing, please interact with us. Please take into account the opportunities. And like here from the Cape Town, we happened to run into nice friends from France-BioImaging because both Sean Salamero and Caroline were there. And so we should look sharply into the future, just like my dog here in the middle of the Finnish snow. I think all good presentations should have a dog. So I couldn't avoid putting the dog here, there. Thanks for your kind attention.
Thank you, John. I think we have time for one question, and then we'll have the coffee break. Yes, Jean?
Are we allowed to translate the poem?
Absolutely. There's no copyright. If you haven't tried out the service, the chat, the AI chatbot, it's a rabbit hole. You will easily get hooked on that.
Thank you again, John.
So, thank you to all the speakers. And just to remind you, after the coffee break at 4, there will be a very short, and don't worry, feedback from the working group. So it would last like maybe 30 minutes altogether. But so please be back at 4.